Hey everybody, in this video I am going to talk about another DNA repair process. This will be the last, I believe this is the last DNA repair process we will be talking about in this course. This one is called non-homologous end joining, or NHEJ for short. So this process is another double strand break repair process. However, it's considered low fidelity compared to some of the other double strand break repair processes that we have already discussed. So essentially, let me just list some of the proteins involved because we may see these names in the future. We're not going to worry too much about the specific details of how scientists think non-homologous end joining works, but there's a specific ligase involved, ligase 4. There's a protein called XRCC4. There are genes called proteins Q70 and Q80, and these proteins are thought to grab onto the ends of DNA after a double strand break, keep them from floating apart from one another. And then we have this DNA PK superscript PS. Some of these are complexes of proteins, I should mention, and there's another one called Artemis. And I just want to mention these because you might see these mentioned in the future, and these are relatively common proteins that you might come across. They're pretty important. So essentially, non-homologous end joining is going to work when a homologous template is not available, right? So some of the homology-directed processes use like RAD51 to help look for homologous sequence in the homologous chromosome to help guide the repair process and copy some of that genetic information over into the broken strand to make sure that the repair is done in a way that will still allow, say, for a functional gene. But sometimes the cell can't always identify a homologous template, and that's when non-homologous end joining comes into play. So if there is a double strand break in a chromosome, that creates these loose ends, and the cell needs to repair these ends. There shouldn't be any loose ends anywhere in the nucleus, because keep in mind, even the telomeres, the very ends of the chromosomes, the telomeres have their ends tucked away in special structures, whether that be like the T-loop or another type of structure called G4 DNA. The telomere, the very ends of the chromosomes, are protected in certain structures because loose ends of DNA are not allowed in the cell. And what happens is we have exonucleases in our cells that look for loose ends of DNA, and they try to degrade that DNA. So if those double-stranded breaks aren't fixed, if those loose ends aren't repaired, then the chromosome is in danger of being degraded by exonucleases. So non-homologous end joining can be used when a homologous template is not available for repair by a homology-directed process. And what are some of those processes we've seen already? We saw double-strand break repair by HR. We've seen SDSA, SSA, and BIR. I think these are 4 homology-directed processes that we have already discussed. So let's take a look and see why non-homologous end joining is considered a low fidelity process. So I've diagrammed some of this for us already. So let's assume this is a DNA sequence on a chromosome somewhere in the human genome. And a double strand break is going to occur in this blue shaded region right here. And let's say that after the double strand break, we get something like this. So we have these overhangs right here. And then we have the nucleotides that were lost by whatever process caused the double strand break. Maybe it's X-ray, radiation, or another process. The DNA is broken, and these nucleotides have been broken from the strands, and now they're lost essentially. And the DNA has these kind of sticky ends here that don't really stick together, right? Because they're not necessarily complementary. So now let's assume non-homologous end joining is going to fix this double strand break. Now I will just mention the Ku proteins, so Ku70 and Ku80 are the proteins that are found in very high concentrations in the nuclei, and they are ready and they grab onto that DNA if it's ever broken. And they're supposed to keep the ends from floating away. So non-homologous end joining can work. So before these can be stuck back together by non-homologous end joining, the ends need to be polished. That means they need to be made blunt-ended. So that can be done in any number of ways, and that would involve exonucleases, polymerases for gap filling. Right? We can fill these gaps with polymerases. Or we can trim away the ends with endonucleases. And then there are also enzymes that can just add nucleotides to the end. So non-templated addition of nucleotides to the end. So we can get nucleotides in here that weren't there before. So down here I have an example. So in this case, exonucleases have just trimmed the ends here. Now we have the blunt ends, and this can be stuck back together by non-homologous end joining. Of course, that's going to involve a ligase, right? So if that's stuck back together, we lost, let's see here, 1, 2, 3, 4, 5, 6 base pairs of genetic information if non-homologous end joining fixes the break in a way that just kind of polishes the ends by trimming and sticking them back together. So okay, we non-homologous end joining fixed the double strand break, but it resulted in a pretty significant deletion of like, what, 6 base pairs. Is that what I counted? 1, 2, 3, 4, 5, 6, yeah. So another possibility is this end could be trimmed and polished that way, and then this TAT right here, well, this could be filled in with the ATA. Okay, and if this is stuck back together, well, what we've lost 1, 2, 3. Okay, we end up with a 3 base pair deletion after repair. So both of these cases, in both of these cases, we've lost genetic information and we have mutated sequences after the repair process. So right away you can see that non-homologous end joining is a low fidelity process. There is a chance, if the break is clean and then there's very little process or no processing of the ends except for sticking them back together, that non-homologous end joining can result in no mutations. But for the most part, the thought is that it usually involves mutation of the area that's been fixed. So let's take a look at another one. Oh, I think that's the only one I've looked at. I only did 2. Okay, but we can get other examples, right? Like I said before, we can get non-templated addition of nucleotides, so inserting other base pairs that weren't there before the break. So there are many different ways to repair this, and essentially it leads to mutation. But it's better than not repairing the double strand break at all, which I implied that it would lead to degradation of the chromosome if that double strand break is not repaired. So, and I said we were only going to talk about one more mechanism. I kind of lied, because I'm going to talk about just one other that is related to non-homologous end joining. It's sort of like considered a variant, or some researchers consider it a variant of non-homologous end joining, and that's called microhomology mediated end joining, so MMEJ. And this is also called, same process, different name, alternate non-homologous end joining. I'm not quite sure which name is preferred by the majority of researchers. But with that process, so let's say the double strand break led to something like this. So we're going back, considering this sequence up here, a double strand break occurs, and the ends, let's say they end up looking like this for some reason. Well, in MMEJ, you can take a look at that microhomology. What happens is we have some pairing between the two ends. So this TA can pair, say, with this AT, and that's the microhomology aspect. So that's the microhomology mediated end joining aspect of the mechanism. And once those are paired, well, then we have these two little gaps that need to be filled by polymerases, and the remaining nicks will be sealed by DNA ligase. So that's all we're really going to talk about with respect to non-homologous end joining and the variant process of microhomology end joining, or alternate non-homologous end joining. Essentially what we need to know is that the cell has ways to take a double strand break, process the ends, and stick them back together. And the mechanisms are NHEJ and MMEJ, and they usually leave behind mutations after the repair. So in the next video, we are going to start taking a look at recombination with respect to antibody production for the immune system. So there's a lot of cool molecular biology going on with respect to antibody production in the immune system. Okay, so see you in the next video.